All right. Thank you, everyone, for joining us for OA Week 2023. This is the North American Hub session. And my name is Kalina Grab. I'm a Canals Fellow in International Policy at NOAA Ocean Acidification Program, and I'm also one of the GOA-ON secretariats. So please note that this session is being recorded. OA Week is presented by GOA-ON, which is the Global Ocean Acidification Observing Network. GOA-ON has 3 sponsoring organizations. First, NOAA, the United States National Oceanic and Atmospheric Administration. Second, the IEA-OAICC, the International Atomic Energy Agency Ocean Acidification International Coordination Center. And third, the IOC UNESCO, the Intergovernmental Oceanographic Commission of the United Nations Educational, Scientific, and Cultural Organization. GOA-ON was established in 2012 with just a handful of members. And since then, GOA-ON has grown immensely. It now has over 1,000 members from 114 different countries. It also consists of 9 regional hubs, which span across continents and oceanographic regions. And we'll be hearing from most of these different hubs and regions throughout the week. If you're not a member yet, you can join GOA-ON today by visiting goon.com, goon.org, or the link that I placed in the chat. OA Week debuted in 2020 and returned in 2021 when events and conferences were postponed due to COVID-19. Following the successful in-person symposium on the ocean in a high CO2 world in 2022, GOA-ON is bringing back OA Week 2023 to maintain momentum around OA research and provide a virtual platform for the ocean acidification community to exchange their latest findings. We are thrilled to present a wide range of ocean acidification topics and speakers from around the world. During the presentation today, everyone is in listen-only mode. You are welcome to type any questions into the question box, which can be found likely on the bottom right of your screen in the control panel. And we'll be monitoring incoming questions and pose them to each speaker after their presentation. Any remaining questions we will answer during the question and answer section, which will begin after the final presentation. For the discussion, you can also use the raise hand function in the toolbox at the bottom of your screen, and we can call on you and you can ask your question directly. So with that, I would like to introduce our moderator for this session, Yumeng Pang. Yumeng is one of our OA Week coordinators this year, so she has been instrumental in making OA Week happen. Yumeng has a PhD in marine biology from the University of Tokyo and is currently a postdoc at the University of Washington, so she is a new member to the North American Hub. As a fisheries biologist, she has been focusing on the impacts of changing ocean conditions on marine organisms and ecosystems and its associations with fisheries and aquaculture productions. Currently, she is exploring transdisciplinary research on comparing OA adaptability of fishing communities between Japan and the US. Yumeng, the floor is yours.
Thank you, Kalina. Hello, everyone. Welcome to the North American Hub session. I'm really happy to be here. And at the beginning, I would like to talk a little bit about the North American Hub session. So the North American Hub is one of the earliest formed hubs within GOA-ON, and it includes over 225 members from Canada, the US, and Mexico. The North American Hub has been integral to GOA-ON, with members serving not only within the hub, but also as GOA-ON Co-Chairs and secretariat members. This session will highlight early career ocean professionals within the North American Hub, demonstrating the diverse and interdisciplinary research that our hub pioneers. So I'm really happy that today we have 4 brilliant speakers talking about their research. And first, I would like to introduce our first speaker. Our first speaker is Patrick Duke. So a little bit about Patrick. During his current PhD, Patrick looks at how the ocean mitigates climate change and is impacted by human emissions. He used machine learning to create high-resolution regional estimates of air-sea gas exchange of carbon dioxide, establishing a baseline of marine uptake particularly in dynamic coastal zones, is important in ensuring carbon dioxide removal is real, permanent, measurable, and verifiable. Patrick is also a sustainability leader in his community, focused on training people to combat climate change through art, policy, and innovation. So Patrick, would you like to share your slides and start your presentation from here?
Absolutely. Thanks so much for the introduction. Hopefully you guys can see my screen there now.
Yes, we can see it now.
Yeah, so I'm going to cover some of the work that I'm doing in my PhD, just wrapping things up here, estimating CO2 source sink dynamics in the coastal zone. So we're mostly working in the Northeast Pacific Ocean off the west coast of Canada and the sort of the northwest sort of coast of the US. And after some work in the open ocean that we sort of wrapped up this year that we published, now we're sort of moving into that coastal space. So why are we sort of looking at this air sea CO2 uptake of CO2 in the coastal zone? Well, policy tends to really focus on electricity, renewable energy generation, coal phase outs, electric vehicles, and the terrestrial sort of biosphere when it's talking about climate change mitigation and climate change adaptation. But if the marine carbon sink is sort of shifting underneath this, then we sort of need to adjust our climate action efforts to account for what's happening in the ocean. And how well do we really know what's happening in the ocean as far as CO2 uptake? In certain regions, it tends to be a little bit different. So this is just from a paper that we published this year where we reviewed essentially air sea CO2 flux values and carbon sort of uptake and outgassing in the oceans around Canada. So we looked at sort of the West Coast and the Pacific, we looked at Arctic, we looked at the Atlantic, the North Atlantic, and we sort of compiled a bunch of different estimates from a bunch of different ways of making these observations or making these estimates from modeling as well. So here we sort of just zoom into the West Coast where these boxes are the coastal ocean. And you can see a couple of different estimates. Each one of these bars is a different estimate of how much CO2 is either being taken up in the blue, the negative values, or is actually being sort of outgassed to the atmosphere in the red or the positive values. And you can see in the coastal ocean, it's pretty variable across a lot of these estimates, particularly on the west coast of Canada. In the Gulf of Alaska coastal ocean, in sort of the Pacific coastal ocean as a whole from some of the more global products that we looked at, and in this Canadian sort of Pacific Shelf region, as well as sort of in this sort of enclosed sea in the Salish Sea on the sort of east coast of Vancouver Island. And when we look at this compared to the open ocean, the open ocean estimates seem to agree quite a bit better. So what we wanted to do was really make sort of another estimate here to sort of help set these baselines for what this marine carbon uptake could look like in this coastal ocean to really help understand sort of future change, as well as validate model output and inform future observations going forward for estimating this air sea CO2 uptake. So how did we actually do that? We sort of went from observations to these marine carbon sink estimates using really what we already had available. So this is a snapshot of 2016, sort of a complete year in surface ocean PCO2 or partial pressure of CO2 observations, mostly from shipboard or ship-based measurements. So a lot of these are cruise tracks from Seattle, right about here, sort of cruising out to Asia markets that all had underway systems on board. And as the sort of ship is moving along, these systems are pulling in CO2, pulling in seawater, estimating how much CO2 or measuring how much CO2 is inside that seawater, then spitting it back out. And we have these sort of cruise tracks, but you can see as it sort of steps through each season, December, January, February here, there's a lack of observations. And then in other seasons, we have sort of quite a few, June, July, August through these steps. But overall, it's hard to make an informed estimate basin-wide about how this coastal ocean is actually taking up CO2 from these pretty scarce estimates. And sort of using a neural network approach, using these estimates that we already have from these direct observations, and then from satellite-based imagery and reanalysis tools, we sort of plug that into the neural network, and we were able to spit out sort of this sort of basin-wide coastal estimate of PCO2 or partial pressure of CO2 across the Northeast Pacific. So how do we actually really do this? The first step is we sort of cluster like regions, or these biogeochemical provinces, or areas that we think sort of behave in a similar way, sort of using a process-based approach with sort of inputting temperature, salinity, sea surface height, and then using a neural network or a self-organizing map to actually cluster these like regions. And this really beats down a lot of the variability that the neural network has to deal with in sort of the next step in estimating CO2 inside of these clusters. So inside of each one of these clusters, in the second step, we take the PCO2 measurements we already have as targets, and then we take a whole bunch of predictor variables from satellite and reanalysis tools, like I said, like temperature, salinity, sea surface height, wind speed, chlorophyll is sort of an estimate of biological activity. And then atmospheric CO2 is sort of a time stamp for as things progress through time. We're increasing CO2 in the atmosphere from anthropogenic emissions. And that's sort of a nice little time stamp for our PCO2 targets in the ocean. And we create nonlinear relationships with these predictor variables and the PCO2 observations that we already have. And then from there, we can sort of scale up and make these basin-wide estimates. And, you know, we're doing a pretty decent job. I think it's pretty tough in the coastal ocean just because the variability is so high compared to what we see in the open ocean. So here we go on to just sort of evaluate. And in this first box, it's SOCAT training data, or the sort of PCO2 observations that the neural network has actually seen along the x-axis here. Along the y-axis is the PCO2 estimate from the neural network. In this first box, and then in the second box along the x-axis is the SOCAT data that we actually withheld. So this is PCO2 observations the neural network has never seen before that we're sort of evaluating against. And the important piece here is sort of this trend overall looks pretty good compared to these estimates. The slope is pretty close to 1. We tend to slightly overestimate some of the lower end PCO2 values and slightly underestimate some of the higher ends, but overall this is a pretty good estimate, and the relative uncertainty relative to sort of the variability that we see in the coastal ocean is actually pretty comparable to a sort of our open ocean product that we developed earlier this year, and a root mean square of about 43 microatmosphere, which is pretty good and pretty comparable to what you see for other coastal ocean estimates sort of globally, especially at sort of high resolution. So here we're at about a 1/12 a degree by 1/12 a degree, which at our sort of latitude is about 6 by 9 kilometers. Yeah, so this is not too bad. But then this was sort of a unique piece that we looked at in the coastal ocean, was really what is this neural network telling us in a process-based way? How well does the neural network really understand what's happening at sort of a process-based level, and not just sort of a, you know, if you see these temperature, salinity, sea surface height values, how does that correspond to a target, but sort of really grouping it regionally. So what we did was we actually used a perturbation-based spatial sensitivity analysis, which is a very fancy way of saying we introduced some noise to each individual pixel in the neural network, sort of predictor variables. So the variables that we're showing the neural networks to sort of train on and make estimates of this approach. And what this really showed was there's sort of 4 distinct tiers of how important these variables are to the neural network making this estimate. And we saw that PCO2 in the atmosphere is actually sort of dominantly the most important control to making this estimate. If you have changed that value, then the surface ocean is very sensitive to what's happening in the atmosphere. And that really makes a lot of sense. And we're actually, from here, we're able to sort of estimate how sensitive regions are, or estimate regions that are more sensitive to this atmospheric trend compared to the rest of the ocean from this sort of sensitivity analysis, which was pretty cool. The sort of second tier of importance was temperature. Temperature sort of directly related to the partial pressure of a gas in seawater. So this makes total sense. You know, it was most of the region is really sensitive to this. But then in this sort of third and fourth tier, where we have wind speed, temperature anomalies, which is just, you know, subtracting the climatology from the temperature values that we're actually measuring with satellites. And then sort of this chlorophyll and salinity values being the least important and not really informing a lot of the estimates. So having this sort of grouping was really important, but then sort of breaking it down spatially was also important for us to see, when and where is the neural network sort of associating these processes to making this PCO2 estimate. And something we found that was pretty cool, here's just the exact same colors that you see in the figure A is in this mapped figure B. We just took out sort of those three most important factors. So temperature or PCO2 in the atmosphere and temperature in the surface ocean. And we grouped these by each pixel that had sort of the next most important variable. And you can see pretty cool in the north sort of gyre region. There's the Alaskan gyre sort of sits up here in the northeast Pacific. And the sea surface height of that gyre boundary really sort of set the tone for what that PCO2 value was. You can also see some eddies here spinning up just off the coast. This is the Sitka eddy along the coast of Alaska. This is the Haida eddy off of Haida Gwaii, Canada. Also, directly related to sea surface height and what we use to infer those eddies, which was pretty sweet. And then sort of down in this southern region, we saw that sea surface temperature anomaly was super important. Here, you don't see a large range, which, once you have sort of an event that's changing this anomaly, it became pretty critical. But then sea surface salinity, really important in these regions in the near shore where you have a lot of sort of freshwater input. So this is sort of the Columbia outflow. This is the Salish Sea with the Fraser River outflow. You can see up here in Cook Inlet, there's another river mouth there and near Anchorage, Alaska. So this was pretty unique and pretty cool takeaway for us, particularly in this coastal zone where we really wanted to see how is the neural network interpreting this sort of predictor variables that you're showing it, and is it able to pull out process-based sort of conclusions to actually make these estimates. But what about the air CO2 fluxes in the region and sort of how is that changing? One of the sort of the pieces that we wanted to look at was just annually, what's the mean annual air CO2 flux across this whole area? And what we found was really in this sort of upwelling zone off the west coast of Oregon, Washington, off Vancouver Island here, we had really strong uptake values nearly year round, which was pretty cool and definitely impacts this sort of net annual budget for air CO2 flux in the region. Another sort of important piece of this was in the near shore, in the Salish Sea, up in Cook Inlet, like I said before, and then sort of through the southeastern Alaskan archipelago, we had a lot of strong outgassing values. And, this sort of contrast between the coast, this sort of near shore coastal ocean, and then the sort of outer coast and along this shelf where we see upwelling between outgassing and then uptake was pretty unique. And then in the sort of Alaskan gyre region here, we also see outgassing, which is similar to sort of some other estimates that are available. So sort of the same picture here on the left, in this sort of figure A, where we have sort of the mean annual CO2 air CO2 flux, blue and the negative values are uptake, red and the positive values are outgassing. But then we also looked at this sort of mean seasonal flux amplitude. So sort of how much the range of the seasonal CO2 flux from winter to summer. And you can see these darker values are higher seasonal amplitudes. And what we found was regions with sort of small mean annual CO2 fluxes, sort of here's a good example here, sort of in the near shore along sort of the edge of the west coast of Vancouver Island, in Queen Charlotte Sound here, sort of on the east coast of Haida Gwaii in Canada. These values where the small annual fluxes are sort of associated with these very large annual amplitudes. So we have these sort of contrasting seasonal CO2 fluxes where it's outgassing in the wintertime and sort of uptake through the summertime where they're actually competing and the net annual flux is pretty close to zero. Whereas along the shelf where we have really strong, large mean annual fluxes, strong uptake values, we actually have pretty low seasonal flux amplitudes. And these sort of competing pieces with particularly upwelling and downwelling cycles seasonally really sort of drive this low seasonal flux amplitude, but sort of strong annual uptake, which was pretty cool and maybe unique to this region with that sort of balance between the upwelling and downwelling regimes that we see along our coasts. The last sort of piece here with the air-sea CO2 fluxes that we wanted to get into was the importance of this nearshore. Great, we showed that there's some strong outgassing in the nearshore, but does this really impact sort of the regional budget for what we're looking at when we talk about coastal ocean air-sea CO2 fluxes, and especially when you talk about sort of these more regional integrated budgets, especially when they go into integrated sort of benthic, marine, or terrestrial to marine, and sort of pelagic water column stock takes for how carbon is moving through the system. Do we really need to include the air-sea CO2 fluxes in this very near shore? So what we did was we looked at sort of another regional product that's available out there from a group at NOAA, Jonathan Sharp. He has this product where we have this overlapping domain with Jonathan's product in the blue along the map, and then all the red points along the map are actually sort of the nearshore coverage that we're adding from our product that haven't previously had estimates associated with them. And then when you look at how this sort of plots up averaged latitudinally, the dashed line is Jonathan's NOAA product, and then the light blue is our product with the overlapping domain from Jonathan's group, so just the offshore. So the nearshore is sort of masked out here. And then the dark line is where we included this nearshore, where we included these red pixels. And you can see that in certain regions, that difference gets quite large, particularly in the Salish Sea. Inclusion of the Salish Sea makes quite a big difference sort of regionally in what the PCO2 does, and then in what the air-sea CO2 flux does here in panel B. But then also up in Glacier Bay, Alaska at about 57 degrees north up here, where the PCO2 difference and the air-sea CO2 flux difference are quite large. But overall, regionally, especially when you include sort of the nearshore past where the Jonathan Sharp's NOAA's product includes in that sort of Gulf of Alaska region further north that our product extends to, it didn't make too big a difference. But when you look at these sort of smaller areas, it definitely does make a huge difference, inclusion of this nearshore. So that's something to consider when you're thinking about these sort of integrated regional carbon flux budgets and whether or not, inclusion of the nearshore is important. So a couple of just key takeaways as we wrap up here. So the neural network spatial sensitivity analysis that we did really identifies the influence of these mesoscale processes, including coastal eddies, freshwater plumes, tidal mixing, and then the Alaskan gyre as well. And this was a really sort of unique test that we hope that more sort of neural network people will use to look under the hood rather than sort of the black box approach of sort of creating these products. And then the coastal sort of Northeast Pacific coastal ocean is a net sink for atmospheric CO2 with large spatial heterogeneity between outgassing in the nearshore and uptake on the outer coast. And then the inclusion of this nearshore can have a significant impact on sort of these regional scale marine carbon budgets. So I'll leave it there. I think we maybe have time for questions. Yeah, that should be good for me.
Thank you very much, Patrick. It's a really cool presentation. We do have some time for question. So everyone, please type your questions into the Q&A box. You can also raise your hand so we can call on your name and pick up the question. So here we have 2 questions for you. So amazing talk. I have a question about the neural network. Do you have a function in MATLAB or R, or how can I get it?
Yeah, so actually with Peter Lenschuster, who developed the approach, it's available in MATLAB, but actually I would encourage you to there's a more accessible version of creating these estimates from Dan Ford at the University of Exeter and Jamie Shuttler. They created a Python toolbox that is essentially plug and play at any sort of spatial or temporal resolution that you want. So whatever you're using for predictor variables, sort of across satellite imagery or reanalysis products, this toolbox can sort of pull all of those products at different resolutions compile them, and put them into the neural network and create an output at the same resolution, same temporal spatial resolution. So it's super simple toolbox. I can post it in the chat maybe after we wrap up, and I think that'll be useful.
Yeah, of course. Thank you. You can type your answer directly, like to the first question, and we have a second question for you. So, what methodology did you use to make a composite map with all the variables?
So we used a feed-forward neural network approach. So the first step was this self-organizing map. That's what we used for the clustering, but then the feed-forward neural network is actually what takes the targets and actually has this output in this gridded format that you see with sort of the composite maps. And we're at a monthly temporal resolution and a 12th by a 12th degree spatial resolution.
Okay, great. And the last question, what is the cause of the high outgassing in the nearshore region?
Great question, Dick. The big driver here, in some of these regions, it's a lot of tidal mixing. So like Johnstone Straits, Strait of Juan de Fuca in the Sailor Sea, we see a lot of tidal mixing, bringing sort of CO2-rich water up to the surface where it's driving this outgassing. But then in sort of other coastal regions, a lot of it is this coastal nutrient trap, where there's just high residence times of this organic carbon in this highly sort of productive coastal nearshore region, that once that carbon starts to respire and stay in the surface ocean, you see a lot of outgassing. And it's hard to make this link with our product alone where it's just PCO2, but we see a lot of this from sort of the Sailor Sea cast model from the Susan Allen and Teresa Yarnikova's work at UBC.
Okay, thank you very much, Patrick. Thank you for your great start of our session. So
thanks so much, guys.
Now let's move to our next speaker, Sam. So Sam is motivated to understand the rules of life that allow organisms to persist in our ever-changing world. As part of his doctoral research, he worked alongside growers in the shellfish aquaculture industry to investigate techniques for hatchery enforcement. Using next-gen sequence data, he explored the effects of early life programming windows when acclimation or priming homeostasis can buffer against subsequent environmental change. Molecular adjustments affecting phenotype are potentially transient, dynamic, and interdependent, but remain poorly understood. As a postdoc researcher at Nova Milford, he used multi-omics data to understand the molecular machinery underpinning environmental resilience within and across generational timescales in bay scallops. So, Sam, the floor is yours.
Great, thank you very much. Let me share my screen. Thank you for the introduction. Hopefully you all can see this.
Yes,
Great.
we can all see that.
Thank you. All right, I'll dive right in. So today I'll be presenting a developmental mismatch of PCO2 levels in a second generation of northern bay scallops. So we just actually finished up an experiment this past year, a massive effort, 3 generations of bay scallops reared under continuous OA treatment. 3 treatments, low, moderately elevated, and high. Our overarching objective for this experiment was to investigate the capacities for rapid adaptation under ocean acidification. And with our efforts, we looked at broad phenotype through physiological, growth physiology, growth survivorship, scope for growth, alongside molecular aspects tied to phenotype, such as single nucleotide polymorphisms and transcriptomics. However, during this experiment, we wanted to sort of add some additional insights into how OA may affect the cell, given that we have full life histories of exposure and across generations. Through this type of experiment, we might be able to disentangle the effects of OA and whether history under environmental challenge can influence how an animal will respond. So the underpinning rationale here is that our fundamental understanding of how OA affects a cell is understudied. What we do know is that gross phenotype is affected, such as growth and survivorship, and early life stages are particularly susceptible. So this gross effect on fitness is a direct tie from the cumulative literature And what we want to understand is, the underpinning effects. Given that these gross phenotypic responses are negative, there's likely an effect on energy metabolism and also cell death that may lead to these gross phenotypic responses. So this alludes to the mitochondria, the energy powerhouse, the underpinning ATP that provides energy for these downstream effects that we see on growth. This was a basis of my rationale for this experiment I'll be presenting today. With our question, are there cellular traits that are associated with phenotypes that are adapted to ocean acidification? We look at model taxa to really give us inspiration for how to organize this experiment, given that the effects of environmental stimuli are often evident by interrogating those that are naive and those that are habituated to that specific encounter of stress. We use developmental mismatch theory, which describes a discrepancy between a resulting phenotype that's induced during development that differs from that allows for an optimal response post-development when the environment changes. So this factors in both the history of the organism and also its subsequent encounter. By looking at an experiment that alters both the history and the encounter, we may be able to disentangle how that stressor actually affects the organism on the cellular level. But we're limited here by model organisms, given that we need continuous exposures throughout the lifetime. Our experiment that I presented earlier, we have 3 generations of exposure of a bivalve, which is unprecedented for that taxon, like mollusks. So we have an optimal opportunity to explore this type of experiment. With our hypothesis that our induced phenotypes exposed to mismatch will reveal cellular mechanisms underpinning OA response. So again, bringing back that multi-generational framework, here just the schematic walking through our 3 generations that we reared under low, moderate, and high PCO2 levels, or a sort of a naive challenge, and then given a moderate and a high challenge. So our resulting adults that I used for this specific experiment had 2 generations under what we deemed as low PCO2, approximate to ambient, and our moderately elevated PCO2. We also had an additional generation that was an additional cohort that had 1 generation under a high treatment. So 3 total cohorts that we looked at for differing histories of OA exposure. So now, so we can sort of dub these in light of developmental mismatch as our induced phenotypes with lifetime exposures to differing environments. Given that we have a naive that was not exposed for lifetime durations to elevated PCO2 and 2 habituated cohorts. And then we gave them a subsequent encounter in a mismatched, fully crossed design, in which the match is just continuous exposure to what they were already reared under, and mismatch is the being given the other 2 treatments for a short period. We measured destructively to disentangle the effects of acute and short-term challenge. So at 24 hours and 14 days of this fully crossed design, extracted live cells from hemolymph from adductor tissue for flow cytometry, and also gill tissues for downstream analyses. As for our measurements, with our live hemolymph, I'm just going to be talking about that today for sake of time. We measured cell viability, so the proportion of live and dead hemocytes, mitochondrial membrane potential of these live hemocytes, and also the production of reactive oxygen species, or superoxide deriving from mitochondria. For our statistical approach, we combined all variables and all factors using a PermaNova. And now to jump into the results. So I'm going to walk through it slowly. It's the full factorial design here, so just want to really kind of walk through it. So I'll be presenting all the results akin to here for both 24 hours and 14 days. All the color on this figure is associated with history. So history being low, moderate, and high. On the top figure, we have the proportion of live and dead cells. Live cells have the darker shading, and dead cells have the lighter shading. On the x-axis of all these figures is the exposure, as L for low, M for moderate, and H for high. The bottom figures, we have mitochondrial superoxide in the bottom left, and mitochondrial membrane potential in the bottom right. The units here are arbitrary given its flow cytometry data. And all the data is as mean standard error. So to jump into the results here, Via our Permanova, there is no effects or interactions for 24 hours. However, I do want to discuss some descriptive patterns that we do see. So the arrow here points from the matched history. So we have high under high, moderate under moderate, and being relieved of that PCO2 challenge, being exposed to a recovery response or a lower PCO2, we see an increase in hemocyte viability, higher proportion of live hemocytes, attributed with a decrease in reactive oxygen species, specifically superoxides deriving from the mitochondria. This gives us insight that a potential relief of the PCO2 challenge resulted in a better fitness, or these potential underpinnings of what downstream better fitness may look like. Interestingly, on the bottom right too, we also see that regardless of subsequent encounter being the x-axis, we see that low history scallops in green had lower mitochondrial membrane potential than those deriving from moderate and high history. Again, these are descriptive patterns. So now to jump into day 14 after a short-term challenge. Here we found significant interactions between history and exposure. And I'm going to walk through the main pairwise terms here. So highlighted in the box, we found where our pairwise terms are significant differences, that low history and high history responded differently to a moderate challenge. And this is a mismatch from their histories, in which those that derived from low history and were never exposed to high PCO2 challenge and were given it abruptly as adults had fewer hemocytes, and those were associated with higher membrane potential, yet less reactive oxygen species. And those that derived from the high history exposed to moderate had subtle change in hemocytes and almost an absent response, as you see as a straight line here with superoxide and mitochondrial membrane potential, no differences between their history and their subsequent exposures. So to really hone in on what we see here as a take-home message, we found that naive scallops seem to be exhibit a change under short-term challenge, in which cells that were able to, viable cells in response to elevated PCO2 have more robust mitochondria, higher membrane potential of their mitochondria. So the second pairwise term that was significant in this interaction were between low history, moderate history in response to high. So also a mismatch, but in response to the greatest challenge. We see a continued pattern as we saw before with the naive individuals having fewer hemocytes, fewer live hemocytes, and those were associated with greater membrane potential. But interestingly, our moderate history organisms or scallops had higher hemocyte viability, yet higher reactive oxygen species. And this potentially points to a poised state to scavenge reactive oxygen species, that their prior history may have allowed for gene front-loading of proteins such as antioxidants to scavenge reactive oxygen species and not succumb to mortalities as we originally saw on day 1 of exposure. So to summarize what we have from this mismatch challenge, we found that the effects of developmental mismatch were evident after a short-term challenge, but not after 24 hours. We found that our naive history cohort exhibited a change in their live cells associated with higher membrane potential. And this potentially alludes to OA causing proton leak and mitochondrial dysfunction, that the cells that were unable to persist or exhibit mortality in response to this sort of novel challenge where these organisms were succumbed to dysfunction. For our adapted cohorts, those that were reared for full life histories under the PCO2 challenge, under mismatch that relieved them of the challenge, they were able to readily respond, increasing hemocyte viability and reducing reactive oxygen species. However, under increased OA, we saw an additional interesting effect in which live cells were associated with higher reactive oxygen species, potentially due to a front loading of genes poised to scavenge reactive oxygen species. So to conclude, we have multi-generational exposures of our scallops, 3 generations, this massive effort, unprecedented for bivalves. However, we found minimal to no change in phenotype. We used this experiment to leverage a knowledge gap, to tackle a knowledge gap in which our fundamental understanding of how OA affects the cell is understudied. And our main question are what traits, cellular traits, are associated with phenotypes that are adapted to OA, given we have gross phenotypes that are that don't show differences in response to OA in our multi-generational experiment, or at least minimal differences. What we found that a naive response to an OA challenge exhibits cell mortalities. However, those that are able to survive are showing robust mitochondria. Those that have high membrane potential are persisting under elevated PCO2 under short novel challenges. Whereas the adaptive response has a baseline higher mitochondrial membrane potential and higher reactive oxygen species, however, does not succumb to cell mortalities when given a PCO2 challenge. This alludes to mitochondria playing an essential role in rapid acclimation and adaptive phenotypes. Our next steps, we have gene abundance analysis that we're going to be tackling constitutive gene front loading in all of our cohorts, and cellular assays to tackle ATP production and oxidative damage. That's all I have for you today. I'll take any questions if you have them.
Thank you very much, Sam. Great talk. So here we have one question for you, and everyone please remember to leave your question in our QA box, or you can now raise your hand and ask Sam the question directly. So here we have one question that is, why did you finish the experiment in 14 days? What would you expect if you continued it for longer?
Good question. All experiments have a time window. So if we could continue it longer, I expect that the responses that we found could potentially be persistent or may have faded with time. So yeah, I think duration is definitely an important factor to increase the resolution. But for our experiment, unfortunately, it was just the sake of a time interval when we could run this experiment, which is not the greatest answer, but.
Okay, great, thank you. Any further questions from the audience now? You can raise your hand. Oh, I raised my hand. So does anyone have another question here? Well, you can also leave your question later. We can come back later when we have the extra time. So thank you very much, Sam. Okay, now let's move on to our next speaker, Ricardo. So a little bit about Ricardo. He's also now doing his PhD in coastal oceanography in the Autonomous University of Baja California. So from an phenotypic, genotypic perspective, he's interested in studying the susceptibility of marine organisms exposed to the co-variables of climate change, especially ocean acidification. He is a data analyst specialized in data sets derived from the biotechnology industry, and he is interested in contributing to human resource training for data science and development of scientific research for new methodologies focused on the interpretation of biotechnology data. Okay, so Ricardo, we would like to say, what do you have for us today?
Thank you so much, Yumeng. I will share my slides today. Okay. Okay, let me know if you can see that.
Yes, we can see it well.
Okay. So hello, everyone. I'm very pleased to participate in the Ocean Acidification Week this year. Today, I'm going to talk about gene regulation mediated by microRNAs in the Pacific mollusk aleatorefescens facing CO2-induced acidification. Let me first introduce you to the significance of our study model, the red abalone. California's coastal waters are home of a multitude of marine invertebrates, including several species of abalone. These genus are marine gastropods that occurs from Central Oregon in the United States to Baja California, Mexico. Abalone has been estimated within the most relevant classes of capture and culture in the worldwide mollusk aquaculture. Today, annual overfishing is resulting in 230,000 tons, which represents an increase of 5 times that the 17th or 2 times more since the beginning actual century. Finally, most recently, red abalone has been listed as a critical endangered species. This is in part because of overfishing, but also because of massive mortalities associated with the early life history of abalone. These mortalities have been associated with the climate change along its habitat. Along the west coast of North American seasonal current drive coastal upwellings of the oceanic waters with high CO2 content and low pH, for which abalone is experiencing at the present day corrosive waters. The thermodynamics of the reaction between CO2 and seawater modified the global carbon cycle in the ocean by decreasing pH, but also the saturation state of calcium carbonate, which is in a mineral needed for various marine organisms to form their skeletons and shells, including abalone. The intensity, frequency, and duration of this corrosive upwellings event is likely to rapidly increase in the coming decades, leaving little time for abalone to develop adaptations. On the other hand, the early development includes an indirect biphasic larval life cycle with a short pelagic period previous to its benthic lifestyle. The pelagic life of the abalone includes the time between the embryo fertilization and the initiation of the metamorphosis, in which various stages of maturation are found until recruitment, including the hatching, legal maturation, and settlement. The larval maturation is completed normally between 5 to 7 days after fertilization, whereas subsequent metamorphosis take place 3 to 4 weeks later. We found the transitory stages that prepare abalone for the adult lifestyle more interesting because it involves complex tune-up into several biological processes intrinsic to cellular differentiation, metabolism, growth, and calcification, which of course can or not be disturbed by the challenge of acidification. Due to the complicated coordinated the gene expression patterns that shapes the development require a regulated gene program. For example, it has been documented a rotation in gene expression activity during larval development. Some genes related to cellular energy metabolism, division, and differentiation repression are activated prior to hatching, but structural components for larval protection after hatching are activated, as well as gene for pelagic benthic transition in competent larval stages. Modeling how development programs are sensitivity to environmental cues can be taken from understand the tolerance threshold at which development fitness is lost, measuring external traits, for example, but also including genetic characterization provides vulnerable insights for exploring biological system weakness or robustness facing ocean acidification. For instance, in other shell-led biphasic mollusks, different range of sensitivity in their fitness was observed, where one of or multiple planktonic species were periodically exposed to acidification waters. On the other hand, a broad variety of molecular pathways in response to these external traits were observed, including shifts in acid-base ion regulation, metabolic process, calcification, and stress response. But regarding this heterogeneity observed, a single unifying principle to predict the impact of acidification in the red abalone could extend to theoretical concepts such as the canalization of the genetic programs. The canalization is a principle design which suggests that the development program are stabilized through different genetic elements to increase phenotypic stability facing stochastic fluctuations. The deeper or robust the walls of these canals, the smaller the change for the program to deviate from its optimum. It has been discussed that the regulation of gene expression can enhance the organismal robustness facing stress. Recently, Samuel Gurr mentioned notably this pathway of plasticity. But in addition, recent revelation into post-transcriptional gene regulation suggests microRNAs can act as key players in the canalizing genetic programs. By interacting with the protein coding gene network and conferring robustness to biological process within the genetic programs of the development. In many multicellular organisms, including mollusks, microRNAs originate as independent transcript from intergenic regions, or also as intragenic from intros or exons, depending where microRNAs originate may represent an evolutionary trait for adaptability. Then after several enzymatic catalysis, microRNA guides the RNA-induced silencing complex to direct the post-transcriptional silencing by interacting with the untranslated region of a protein-coding gene. It is well known that microRNAs confer accuracy and uniformity during developmental transition, but also can buffer the transition noise, the transcriptional noise associated with various physiological processes in marine organisms facing environmental stress, including acidification. So what we think based on the mentioned clues, first, regarding the projection for natural abalone habitats, determining its sensitivity to acidification during its early development is an urgent task. Note that although the effect of the sudden acidification or short-term involving times of spanning hours has been demonstrated in many planktonic mollusks, the literature today does not reveal any clues regarding the effect on prolonged periods of acidification or long-term involving the time spanning several days, nor the prevalence of this deleterious effect throughout the pelagic periods. So, understand pathways of stress response, for example, acute or chronic, provides vulnerable insights for exploring red abalone vulnerability. Therefore, we think to evaluate the critical early life history stages of red abalone, contrasting stress response under 2 levels of CO2-induced acidification. Finally, the stress response under short and long-term tolerance can be evaluated during the settlement. Because gene programs may be disturbed by the challenge of stress, assess external traits, and include post-transcriptional gene regulation mediated by microRNAs characterization provides insights about plasticity and canalization of the developmental process in the covariates of acidification. Therefore, we set up a CO2 acidified water-based system to achieve the control of dioxin carbon in seawater. A set of 2 water flow systems was assembled. Here, independent pH controllers were calibrated using the National Bureau of Standards and configured to maintain the water of the desired pH to experiments to moderate and low pH levels respectively. The pH, dioxin carbon, and aragonite saturation state was calculated from dissolving organic carbon and total alkalinity. Moreover, morphology, developmental success, and growth, aerobic metabolism, and calcification, including birefringence and scanning electron microscopy, were evaluated during several stages of the early development. The relative effect size also was calculated to normalize the response radius during the assessment. Here, as decreased the assessed values was negative the effect radio. And finally, molecular approaches were assessed by sequencing small RNA libraries from larvae samples from the hatchet and competent larvae stages. The present research showed results that are in concordance with those in the literature regarding the early life history of abalone. Several early life history stages revealed a prevalent negative response in the calcification, growth, and developmental rates during experiments that mimics 2 near-future acidification scenarios. Moreover, the disturbance observed was stage-specific, suggesting that the body abilities evaluated are at the expense of their developmental stage. This stage-specific response is in fact in concordance with perspectives about windows periods in ontogeny, in which individual stages experience a particular sensitivity to cues on the developmental programs. During the settlement, signs of shell deformation and microfracture were observed in larvae reared under acidification in comparison to normal shell from larvae reared in normal conditions. But despite the deleterious effect on shell structure, the settlement suggests that these larvae were resilient to different scenarios of chronic acidification because no difference was found. Regarding the hypothesis of differences in the chronic and acute performance response, we observed that the settlement from larvae reared to acidification prior to settlement were significantly decreased, although shell deterioration was not observed. So in conclusion, larvae from cumulative acidification stress showed shell deterioration that did not affect its settlement, but larvae related to acute acidification did not show shell deterioration, although the settlement success was affected, which suggests that vulnerability is limited to the difference by weight of response, even stress and levels of acidification in the abalone early life history. Oxygen consumption at 24 and 110 hours contributes significantly to the increase in the metabolism during the larval development compared to the pH 7.6. This altered physiological performance was opposite with a decrease in the proportion of hatchet and normal larvae, as well as competent larvae observed, suggesting that an additional energy requirement is needed to satisfy the genetic program of larvae development at the expense of the observed negative effect in the parameters evaluated. More than 200 million reads were sequenced on the Illumina platform, and after bioinformatic analysis, 147 microRNAs were identified encoded in the abalone genome, among which 75 ones were known and 72 ones were novel. The novel microRNAs might be transitory or specific in abalone during development because they were not similar to any reported mature microRNA in the databases used, but abalone genome. On the other hand, a rotation in the microRNAs mediated canalization in the development program were observed. Under normal pH, high expression levels of several microRNAs associated with regulation of the log system in abalone were found during 24 hours, contrasting to the competent larvae during 110 hours, where several microRNAs associated with the regulation of the pelagic benthic anticipatory transition were observed. Opposite to low pH, where expression levels of several microRNAs usually expressed during 24 hours was delayed or down expressed but overexpressed until 110 hours. Finally, change in gene expression suggests that microRNA's regulatory network is modified in response to ocean acidification. Here, several biological processes were deactivated by microRNAs, including differentiated process from the developmental program. This process may also be associated with transcriptional noise or molecular pathways, and phenotypes mentioned before are disturbed by the stress of acidification. In conclusion, abalone larvae show a stage-specific reduction in growth, calcification, and ontogenic development in response to acidification challenge, resulting to be severe under pH 7.6, although no lethal, because in both challenge, larvae were able to complete the planktonic benthic transition through settlement. Additionally, respiration rates were amplified due to stress, suggesting an unnecessary physiological cause to maintain cellular homeostasis in response to acidification covariates that affect growth, calcification, and the ontogenic development. For first time, the regulatory network of microRNAs that are canalizing larval development of red abalone was identified. This network of microRNAs was modified by the effect of acidification. Here, genes associated with growth, skeletogenesis, immune system development, neural and tissue differentiation are downregulated in response to acidification. This negative regulation is consistent with the reduction observed in the phenotype variables evaluated. Finally, the present research contributes to the initiative proposed to understand carbon cycle and its interaction with marine resources in the Mexican oceans. Before I finish, it's important to mention this work was possible because the collaboration of many people presented here. Thank you very much for attending my presentation. Any question, feel free to contact me.
Thank you very much, Ricardo. It's a really great presentation. Let's see if we have any questions from our attendees now. You can either type your question in the QA box or you can just raise your hand now. So may I ask a question?
Sure.
It's really cool, and also I do similar kind of research about oyster agriculture. So I'm really interested in the abalone agriculture in the local fishery. So I'm kind of interested in like the ocean acidification adaptation part. So I wonder, like through your research, like how do you think that we can adapt to the ocean acidification, like based on the result from your experiment on abalone's early stage? Like what kind of measure or what kind of action that we can take to protect the abalone from suffering from the attack of ocean acidification?
Great. So nice question. Maybe taking in account how acidification is disturbing the early development of abalone can establish some concepts to improve the culture of abalone in aquaculture. I don't know if this is answer more or less to your question.
Okay, and
because,
I kind of yes.
Because acidification in some stages impairs the development, but in other stages it looks like improve the development of the abalone.
Okay, yeah, that's true. It's kind of like a contradictory in some stage of the abalone. So I was thinking, how can we like, like take advantage of like this situation to help abalone to grow, and but also in the same time to protect them from the further damage.
Sure, and this heterogeneity in the response is connected to the molecular and cellular point of view of which mechanisms are activated during different stages and are buffer in some way the stress via the acidification. So.
Cool. And I'm kind of curious, like, so in your experiment, you use like a 2 pH level. One is like 7.6, another is like 8.0, if I remember correctly.
7.8.
Okay, yeah, sorry, 7.8 and 7.6. So I wonder how about like the pH level in the local water, like in the local water, what is the pH level in the natural environment? Why did you choose like 7.8
Yes.
and 7.6?
Yeah, today there is fluctuations of pH ranging from 7.6 to 8.
Okay.
And this is the actually a condition that abalone experiences, but surface pH are actually the pH of 8, but fluctuation remains occurring.
Okay. Thank you very much, Ricardo. Thank you very much for your presentation.
Thank you. You're welcome. So I go to.
Okay, now let's move to our last but not the least speaker. So Claire Botella. So Claire Botella is also a PhD candidate in the statistics at the. Sorry. So Claire is also a PhD candidate in statistics at Dalhousie University, working with a lot of co-workers like Douglas Wallace and etc. So their research uses the statistical methods and the time series techniques to estimate the spatial temporal variability of the carbonate system in the North Atlantic Ocean. So Claire, are you ready to share about your research with us?
Yes, thank you.
Great.
So I'll just dive right into it. And after that great introduction, so with the motivation of my work is probably something you're similar, like you're used to, with atmospheric CO2 increasing. This is causing the CO2 in our ocean to increase and then causing ocean acidification, the main point of our week. So in the Northwest Atlantic, this is an important area because it's one of the main carbon sinks. But unfortunately, there's a lot of sparse observations in this area through time and space. So through today, I'll be analyzing the Labrador Sea within the Northwest Atlantic. I'll be looking at the carbonate system, and ocean acidification is often measured through pH, but pH is part of this carbonate system that includes dissolved inorganic carbon, DIC, total alkalinity, TA, fugacity of FCO2, which is related to PCO2, and then finally pH. So in literature, we've often found that direct observations are very sporadic, so we end up doing these spatial climatologies, but there's very few analysis that have done time-focused measurements. So today, I'm hoping to try and really emphasize the time trends of the carbonate system and how they are changing for each of our carbonate variables. Analysis of these variables have also been done very individualized. So pH is a strong focus, but then we've also looked at air-sea flux in the surface. But I want to look at this multivariate system with the carbonate system. We're going to do this time series analysis that is going to use a data fusion of multiple data sources and try and analyze all four at the same time to try and by doing them at the same time, you'll see we're going to try and do this data augmentation where we're actually increasing the amount of data for pH, even though it's not direct data. It's going to be calculated data using CO2 sys, which is able to calculate these chemically balanced variables. So the data I'm using is going to be these three data sources. We have GLODAP, SOCAT, and Argo. GLODAP has a lot of the variables, DIC, TA, pH, and some FCO2 for many years and for the full water column. For SOCAT, it focuses on FCO2 in the surface. And then BGC Argo is giving us pH, and it's only for the recent years. And in this specific region, it's only since 2018. So for our analysis, we're going to be taking these observations and averaging them by month and depth layer. So I'm using this statistical state space model, and it gives us a framework to sequentially estimate our carbonate system variables through time. I have the equations there, but the plot is really going to be describing how the equations work. So our state is our black line, and we're going to try and estimate the state of our variables through time. And as an observation appears, we're going to update that estimate to be closer towards those observations. As the observations start declining, then so is the estimate of pH. So this plot very clearly seems to show our ocean acidification. On this plot, there's also red dots, which are direct observations from GOA-ON, and it would also be from Argo, but not in this time frame. But I also have these X's in blue, which are these pseudo observations that I've calculated using the other variables. So the full carbonate system we're showing here, we have DIC observations, and then when a DIC observation is available, we also pulled out the alkalinity observation, and using those two, we've calculated pH using CO2 sys. Because of this, our four variables actually are going to be drawn to vary and stay in this chemical equilibrium state, at least momentarily. And then, of course, through time, all the observations are going to update it. So this is our method overall. And now we're going to focus on these lovely colorful plots. So here I just wanted to show you temperature because to get us acclimatized to what these plots are showing us. So on the right, you see the colorful heat map where from left to right, we have time increasing from 1990 to 2016. And then from top to bottom, we have the depth. Each row is actually representing a time series as shown on the left. So the heat map is showing that the surface has high variability and also higher temperatures than the deeper waters. With all that variability, we have that thermocline going deeper in the winter. So there's lots of features we can draw from this plot. Now for the carbonate system. So we have DIC, FCO2, TA, and pH, and we'll focus on them more in more detail later. But I just want to show you that this is the output from that statistical model. And there's so much detail embedded here. But one thing that's interesting is how like DC has that seasonal cycle actually, as well as like a 4 to 5 year cycle in the surface layer, and then it's much higher DIC in the deeper waters. But then for pH and FCO2, we have a much more gradual change of both of them for all depth layers. I know a lot of interest is on TA versus NTA, and while there's lots of these variabilities in TA, we wonder how much of that is related to salinity. So when we normalize that to remove the effect of salinity, we find that NTA, the normalized TA, actually just has a lot of variability that's not statistically significant. All of these variables have errors, and we can definitely talk more about that later, but the errors, the point right now is just to say that with the statistical method, it's very rigorously estimated errors to go along with it that are varying through time and space, depending on how many observations are available. From our results, we could also then calculate other aspects of the carbonate system, such as separating DIC into its species, the carbonic acid, bicarbonate, and carbonate. And we can also calculate the Revel factor, which is often used to estimate anthropogenic changes over time. Now focusing on pH, which is what we're looking for, and to see how ocean acidification is changing over time and depth. So in the near surface, those top 10 meters, we have this steady decline of pH at a rate of about negative 0.0026 per year. In the time series on the left, we see how the method is going through multiple types of observations from the GLODAP, from the calculated pseudo CO2 cis observations, and then also BGC Argo at the end. If we look at the deeper waters, then all the interior ocean is changing at similar rates, slightly lower at negative 0.003 units per year. But then when we look more closely at these time series and the effects of BGC Argo, we find that we need to do more quality control on these observations, because what is a declining trend through time suddenly is having a different time series characteristic. So this is the method. There's two parts here. So I really doubt that there's this increasing trend just at the very end, but it is showing that this method is very adaptive to the observations given. So so you can very reliably trust that if there is this anthropogenic change happening in the ocean, this method is able to pull it out of the observations and show it in its estimate, which is the black line. Unfortunately, it's also very susceptible to poor data. So for here, I can increase the errors associated with certain observations if we don't trust them as much. So I specifically did here because I wanted to illustrate the point of how it is really tied towards direct observations. On here, I've also shown the aragonite saturation horizon, and below this horizon, it's undersaturated, and this is causing the dissolution of shell-bearing organisms. This horizon's increasing over time at a rate of up to 24 meters per year. So it's shallowing from about 240 to about 800 meters over the 33 years of this analysis. This is a similar estimate to others that have predicted in the region that anticipated it to be shallowing about 700 meters by 2050. This is also a much faster shallowing than in the North Atlantic than is in the Pacific, which is only shallowing about 1 to 2 meters per year. If we look more closely at our uncertainty, so here I've shown the uncertainty of pH. And I know there's lots of colors, and it's really just showing right now where observations are and how much they're trusted. At the very beginning, before 1995, there are very few direct observations of pH, and there's also very few pseudo observations of them. So there's a high uncertainty in this region. Afterwards, we have the blue color, so we're much more certain, and the uncertainty has decreased. It's down to being 0.006. And then after 2018, we have BGC Argo, so it's dropped even more just because there's a lot of data. But if we increase the uncertainty of those Argo data, or we better yet is if I improve the quality control of the data we use, then we see how the uncertainty is changing with the observations availability. The other thing that's tied into this is the variable surface also is going to have a larger uncertainty. When we compare our uncertainties to other methods, we're saying that our method with this time correlated statistical model has uncertainties that are about 60% smaller than if we did this simpler time independent method. And in reference to the time independent method, that's where if we just took all the observations independently and run them through CO2 sys. So we are doing this, the statistical method is actually helping really to fill in those gaps between the observations. And it's not only helping pH, it's also helping FCO2, which has very few observations in the deeper waters, only in the surface waters. So to summarize, this is a very methods approach, but it does have very useful impact on like how to track and how to use the data that is very sparse in this region. Overall, our objective was to estimate the carbonate system and its carbon inventory over time and depth in the Labrador Sea. And we used a multivariate method that created this monthly time series of our variables. And it also was able to amalgamate multiple data sources and effectively creating this data augmentation for some of the variables like pH and FCO2 that had very few direct observations of themselves. By using CO2 sys to do these calculated augmentation of data, it's also meant that these four time series are actually chemically balanced, at least at these moments in time. And by doing this time series correlated method, we've also managed to reduce the uncertainty of our variables by 30 to 60%. By tracking this through time, I think a next step would be interesting to see how not just an absolute pH is affecting the organisms in the ocean, but also potentially the rate of change, if the rate of change of pH also has an influence on the organisms in the ocean. Thank you.
Thank you very much, Claire. It's great, very great presentation, and we're waiting for some attendees to see if they have any other questions. So if you have any other questions, you can type it in our Q&A box, or you can just raise your hand and ask Claire directly now. Let's see. Oh, here. So Patrick have a question now. So how about Patrick? You can ask the question now, so we can wait for others in the meantime.
Yeah, for sure. Great talk, Claire. Great presentation. I had a couple questions. I guess the first one was about the TA signal and the trends that you saw in TA. And I guess it makes sense to me, the deseasonalized trends, but is there a spatial component to that? Or the salinity normalized trends? Like, is there a spatial component to that? Like when you get closer to the coast, do you see any changes just because of glacier inputs or how that's changing?
So specifically in this analysis, we've removed the coastal region because that's probably an issue. And one thing we actually found is we had to separate the North Atlantic and the lab C and emergency separately because there was also these differences with TA between the two regions. So there definitely is a big spatial component. And one challenge with this is we're trying to pull out more of the time trends, but like you have to average spatial like a certain amount to get enough data to do anything. So trying to have a big enough region but not have the confounding spatial thing is definitely this like trade-off challenge.
Right, that makes sense. The other piece was the DIC subdecadal trends. That is super cool, like the 4 to 5 year trends that you see in those cycles. But it's weird that it's not reflected in the PCO2 or in the pH. Is there like a guess why?
I'm still thinking about it. When in previous work, we were doing the larger North Atlantic region instead of just the Lab C, and the 4 to 5 year trends weren't as prominent in DIC. The seasonal cycle was much more prominent, but also the method was slightly different. We pulled out the seasonal cycle and things like that. Here we didn't. So I think a little bit is the DIC just has such a prominent seasonal cycle that pH doesn't as much. And then also well, correct me if I'm wrong. I don't think it does. And then I think one thing that's also just like very different between DIC and the other two is that like DIC's range over the column is just so different. And the surface, it is much lower and has that big seasonal cycle, but it's so high for so much of that water column that it's hard to like notice any of those smaller trends versus FCO2 and pH both have this like smaller region. So it's just, I'm not answering your seasonal cycle question, but like they do seem to be on different, just different amplitudes. So
Yeah.
the fact, yeah, more data is always the answer, right?
It's cool. No, those subdecadal trends are super cool. And then the last one was that, do you have any recommendations for observational people about that? The pH, you showed that uncertainty in pH plot. And it totally makes sense to me that, earlier, those pH measurements, the uncertainty is way higher in the surface and through the water column. But then there's that little pocket in like the lower, below 2000 meters, where uncertainty actually increases in time, at least from a certain point, like from a reference of like 2010 or something. Is there some recommendations? Is that like, do we need more deep Argo to resolve that? Or do you have some observationalist sort of pointers to bring that down?
Well, so the reason why there's this like high right after 2018 is just because Argo doesn't go deeper than 2000 meters. So it's basically this plot just really tells us where we have data and where we don't. So it's, if that uncertainty is fine, is of a level that's okay, then like you don't need the data. But I think one thing that's still unclear is like what level of uncertainty is useful, or like at what point is uncertainty hiding behaviors, and at what point and like how much smaller can we even go? So that is like a sensor question. So I know I definitely really enjoyed that session the other day. But specifically this region is just because there was no data. So that's why the uncertainty just skyrockets when there is no data. So I think, I guess the outcome of my work is really regular data of some sort is useful.
Okay, thank you very much, Claire, for your presentation. And thank you again, for everyone, pretty awesome presentation. I really learned a lot from all of you. And now I will turn over to Kalina to wrap up this session. So Kalina, please.
Yeah, so thank you to all of the speakers today and the organizers of this session. I also thank you, Patrick has put a link in the chat for the Iconic community, which is the early career community in GOA-ON. So if you are interested in joining, you can click on that link. I also wanted to make an announcement for the North American Hub that there will be a in-person North American Hub meeting on January 17th and 18th in 2024, so just a few months from now. This will be hosted by Dr. Wei-Jun Chai at University of Delaware, and we are still working on the final agenda, but some of the topics that will be covered include the new terms of reference for the Hub, which if you're on the Hub mailing list, you will receive emails about that, and you can offer input currently to the draft of that. And I'm putting the link in the chat. If you would like to join that emailing list, you can join GOA-ON and select North American Hub. We will also talk about Hub objectives and collaborations, as well as need-based assessments, best practices, emerging technologies, MCDR, reference materials, and knowledge to action plans. So we really hope that many of you will decide to come join us in person so we can gather as a hub to talk and meet and share our findings and knowledge. If you have any questions, you can feel free to email GOA-ON Secretariat, which will come to myself as well as the other two secretariats. And you can also email us if you're interested in attending. Soon, we will have more registration details and a more formal registration coming. So with that, I would like to close this session and thank you, the audience, for joining us in OA Week. And we please do not forget to consult the website and to register for any other sessions that may interest you. If you would like to stay up to date with the GOA-ON community, consider signing up as a GOA-ON member if you're not one as well. And again, you can select your regional hub, such as North American Regional Hub. And you can discover more about ocean acidification research for sustainability. You can scan the QR code on the left. So with that, I will stay on for the next 2 minutes in case anyone has any questions about the North American Regional Hub meeting. And otherwise, thank you to all of the presenters in this session.